Hi, my name is Shannon Stewart.
Hi, I'm Sarah Lord.
Hi, I'm Alexandra Frias, and this is a presentation for Bio 161 on Fragile X syndrome.
So, Fragile X syndrome shows up very early in childhood development, and it has the same signs or symptoms, as you could call it, as a lot of the autism spectrum disorder syndromes or symptoms, sorry. So, some of these symptoms are slow childhood development, which could be sitting up later, rolling over later, talking later, walking later, later being later than normal. Social behavioral troubles. So that can be crowd sensitivity, noise sensitivity, all of that. And then learning disabilities as well. So that can be reading later, speaking later again, just general learning disabilities. And then Fragile X syndrome can also show up in the physical, in a physical manifestation. So some signs are large ears, long face, and in men after puberty, larger testicles. So most females that have this disease show little to no signs, but it's more apparent in males. And it affects the brain. So we'll talk about it later, but it doesn't allow for a protein to be created, so the brain doesn't talk to itself as well as it should. Um, so it is a sex-linked gene and is on the X chromosome, and it is recessive. It's specifically located on the FMR1 gene, which is on the X chromosome again, and specifically the CGG segment of that DNA. So males and females can carry it and pass it on. So if it's an intermediate in the mother and she passes it on to her child, it will most likely turn into a permutation. However, if the father has an intermediate phase or stage, meaning not as many replications of this segment, it most likely would not be turned into a permutation. However, it is the same way with a permutation. So, as it's inherited by offsprings, if it's a permutation in the mother, it will most likely turn into a mutation in the offspring. But if it's a permutation in the father, it most likely will not turn into a full mutation in the child.
Now for the cause of Fragile X syndrome. So, Fragile X syndrome is caused by a mutation in the FMR1 gene, which ultimately prevents synthesis of FMRP, an important protein that maintains connections between brain cells and the central nervous system. So, what happens is normally the FMR1 gene contains a repeating sequence of nucleotide base pairs, CGG, but in Fragile X syndrome, they are repeated an abnormal amount of times, which causes the gene to be unstable and turned off. When this gene is turned off, the protein FMRP cannot be synthesized. And the lack of this protein is what causes the adverse effects of Fragile X syndrome. For the treatment of Fragile X syndrome, there unfortunately is no current cure for Fragile X syndrome, and the most effective treatments involve symptomatic treatments such as therapies which parallel autistic therapies that help kids learn basic skills that they would otherwise know, or there are medicines that help with behavioral problems, but these don't exactly hit Fragile X syndrome at its source. Something that's being explored is the reactivation of the FMR1 gene, and there's a number of different hypothetical ways to do this, which are shown in the picture below. Each letter is like a different way to reactivate the gene. Most of them are either not effective in their current form, or they involve an agent which can be toxic to the body in other ways. The most effective ones that could possibly provide a cure for Fragile X syndrome in the future are under letters C and E in the picture. Letter C is the application of non-coding RNAs, and letter E is CGG repeat deletion via the CRISPR-Cas9. So these would hit Fragile X syndrome at its source. And would allow the FMR1 gene to produce the FMRP protein.
Now onto the diagnosis of FXS, and it's through a blood test called the FMR1 DNA test. And it determines a specific gene called the Fragile X mental retardation 1 gene, or the FMR1 gene for short. And it looks for a specific number of codons in the beginning of the gene, CGG nucleotides. Those who have 0 to 44 of these codons are considered typical. Those who have 45 to 54 are in the intermediate level. Those who have 55 to 200 of these codons are premutation, meaning they can be carrying the disorder. And over 200 of these codons means full mutation having the disorder, which results in the gene turning off, stopping the production of proteins for proper brain development. As you can see, this does play a big role in the central dogma. There is genetic counseling, however, it is very complex since they have to determine those who are premutation and those who are full mutation, which is a quite number of individuals in the family. Now, who has the disorder? Well, it is inherited, it is X-linked dominant, and it occurs in one of the two sex chromosomes, the X chromosome. And it occurs in 1 in every 7,000 males, 1 in every 11,000 females, and 1 in every 249 females carry it. As you can see, it is way more common in males. I chose this image because it shows a pedigree of a typical Fragile X family tree. And it shows that most of them actually carry it, and some of them have the full mutation. This pedigree also shows a repeat size of the CGG codon. Those who have under 100 repeats either carry it or don't have any involvement with the FXS disorder, and those who are way above 200 actually have the full mutation. This is diagnosed between the ages of 35 to 42 months old through a blood test or through clues such as a broad forehead, an elongated face, large ears, and in most men it's larger testicles. Now, there is this very interesting article on the NCBI page on Fragile X syndrome disorders, and it does go everything in depth, like its genomics, its heritability, its characteristics, diagnosis, and everything. And something we did find very interesting is how the FMR1 gene from the DNA is transcribed to mRNA and translated to a protein that affects one's brain development. This is our presentation on Fragile X syndrome, and thank you for watching.